Hey, welcome to the Seeing Eye to Eye podcast. Today is a mini episode, a little different than our long form episode. Don't worry, we have plenty more of those to come. Today, we just wanted to do a little medical minute here, talk about two big advances in the field of medicine that likely will have implications for you, your children, grandchildren down the line. I'm going to kind of steer the ship today. The two topics are one, CRISPR-Cas9. What is that? I'll go over that. And two, Neuralink, or the brain-computer interface, or BCI. What is that? Again, I'll go more into that. Thoughts from you, Shannon, so far?
It's all you. All you,
Okay,
Max.
so I will direct the attention. I'll send these slides over to you so that our viewers can see them.
Yeah.
And a lot of these slides, I have to give a shout out to a great YouTube channel called Professor Dave Explains, where he goes through these in greater detail. We're going to kind of be more superficial, but this will guide us. Just to take a time machine back, scientists started to notice that bacteria seem to have their own immune system, which is kind of crazy if you think about it. They're these single cell, very simple organisms, and yet they could fight off viruses that were infecting them, and no one knew why. Humans, we have very advanced immune systems. We have an innate immune system where it senses a threat, it's a very generic response to that threat, and then there's an adaptive immune system that identifies a threat, characterizes that threat, and then specifically targets that threat. That's what you see with like immune antibodies, B cells, that kind of stuff. So this seemed to be more on the innate immune side, more simplistic version, but it doesn't mean we understand it, right? So they had to do some science. And what they found was in a lot of bacteria, there are these regions of DNA that repeat themselves. And the pattern is this specific pattern. It's a palindrome. Like think about the word race car, right? Race car is the same way forward as it is backwards. That's a palindrome, right? So they found these palindromic regions, and they're like, oh, this is kind of weird. Why do they have these? And why is there DNA that's not palindromic in between that kind of looks like virus DNA? They kept looking into it, and they brought it all together and realized that the bacteria had special proteins that would go snip little sections of infecting viruses and implant that into its own DNA as sort of a memory bank of things that had attacked it in the past. By the way, bacteria can share their DNA amongst each other, right? So there is a way to sort of spread this knowledge with your buddies so that you're not all taken out at once by a virus. And so in effect, again, they take these sections of viral DNA and put it into their own DNA and then use it sort of like the way police use photos of criminals to try and identify them out in the world. So have you seen so-and-so, ma'am? And they show you the picture and she goes, yes, that's the guy who stole my purse. Then they know who stole their purse and they go and arrest them as an analogy. In this case, it's the cell going out, attacking that viral DNA and chopping it up and destroying it. Okay, this slide 5, is demonstrating the CRISPR system. What is CRISPR? It's clustered, regularly interspaced, short palindromic repeats, CRISPR. And then Cas is CRISPR-associated sequences or protein. So basically, there's a strip of DNA that repeats itself, and then there's a protein that works with that in order to fight these viruses for the bacteria. I want to draw your attention to this slide. So this is slide 8, just showing the mechanics of that, where the bacteria takes the DNA, turns it into RNA, and it's sort of its mug shot. It goes up to the DNA, takes a little sample, sees if there's a match, and if there is, chops up the DNA of the virus and kills it right on the spot so it can't get infected and destroyed. So the genius here was two researchers, Dr. Doudna and Dr. Charpentier. I might be pronouncing that one wrong.
Slide 10.
This is slide 10. Met each other in Puerto Rico at a meeting in around 2011 or 12, and they decided to work together and try to figure out this mystery. What's happening? How do bacteria actually do the thing that I just told you about? And they pulled it off. And they also realized something else. Instead of just using this for research purposes, maybe I could take this same idea, chopping up DNA, and use it to help people. How do you help people? Well, if I deliver the CRISPR system with that protein and a mugshot, so to speak, to go find a gene mutation in your DNA that's causing you some sort of illness. It could go in there, remove that defective piece of DNA, and replace it with a new piece of DNA to help you function better and live a healthier life. So that was their genius. And they were rewarded for that with a Nobel Prize. And actually, fun fact, this is the first time, I think this is still true, that it was an all-female Nobel Prize in medicine and physiology. So there's amazing stuff on YouTube. You can watch and learn about them. They seem like really great people and very smart. What are the bigger implications for this? What are specific ways you could help people? One is gene editing for embryos. Now, this might be controversial, but let's say you found out that you have a mutation in your genes and you're concerned it may pass along to your children and cause them harm in some way. You could get in there and deliver a gene therapy to help them. So yes, so basically we could help prevent kids from being born with certain illnesses. Again, this is sort of controversial and we don't have to get into that, but that is a potential. Another one is HIV. This is slide 12. HIV, in case you're not familiar, is a type of virus that can insert its own DNA into your body. Basically, it becomes a part of you, so that even with the medicines, these amazing medications we have nowadays for HIV, those really suppress the virus from growing more. But even if you get rid of all the virus that's floating around, there's still some trapped in your DNA, and it can come back out at any time in the future. So why don't we get in there and take it down? And so you would send CRISPR-Cas in with the mugshot of HIV, goes into your cells, identifies HIV DNA, snips it out. You no longer have HIV. That's a potential here. Another slide, slide 14, demonstrates perhaps a potential for plant engineering. There's an acknowledgement that water is becoming more scarce in some places and less scarce in other places. And there's traditional crops in these areas that are used to a certain amount of water, right? Perhaps we could gene edit these plants to either be able to handle more water or handle less water or deliver more nutrition. In fact, there's a podcast we both listen to called All In Podcast, where one of the main hosts runs a company that makes this kind of product. They mutate plants to generate more nutrition and make them sturdier or hardier for tough conditions. So this stuff is happening, whether it's with CRISPR or not. There are other ways. Okay, let me see if there's anything else. Okay, and just before we move past CRISPR, I just want to point out again that this has been done before. CRISPR isn't the only way to deliver gene therapy. In fact, the first gene therapy ever approved by the FDA in the United States was for eyes, ophthalmology-related issues, specifically retinitis pigmentosa, which is a terrible eye disease that leads to significant vision loss. And if you happen to have a particular mutation, RPE65, this is slide 19, by the way, they found they could inject a virus carrying a little circle of DNA called an episome, and the virus could infect your cells and then deliver that little circle of DNA, and your body will read that circle of DNA and make the protein that you're missing out on and help you see better. This medication is called Luxturna.
Did it work?
It does work. There are amazing videos. I don't have them on hand right now, but if you go on YouTube, you can see these folks before treatment. They do this test where they have them walk in sort of a dim room with a lot of trip hazards and turns and stuff like that. It's not super complicated, but if you can't see well and it's dark, that would be a challenge for you if you have retinitis pigmentosa. And they time them and then count the number of times they run into walls and stuff like that. And then they came back after their treatment and repeated it and compared their times and their number of hits between the two. And it's like breathtaking to watch these people get back that mobility. Now, are they going to be able to read super fine print? No. But they're less likely to fall, they're less likely to trip, and they feel more comfortable in the world, which is a win from my perspective when you had no hope before. This is slide 24. This is a video of Luxturna being injected. It comes in a fluid with the virus in it, and you do retina surgery where you go into the eyeball and put a little cannula underneath the retina and inject that fluid, which contains the virus, under the retina. That fluid bubble then disperses in what's called a bleb or a bubble under the retina and infects the cells on purpose. We're purposely infecting cells. but with a harmless virus that delivers the gene that these folks need. And to kind of wrap it up, what are other gene therapies in existence? I went to the Children's, this is slide 25. I went to the Children's Hospital, I think this is CHOP or Boston Children's website of available therapy, since most of these things affect children, unfortunately. The most recent ones were Casgevy and Lyfgenia. These are both for sickle cell disease, which is a terrible disease where your red blood cells don't function the normal way. And instead of being nice discs, they turn into these sickles, or like sort of a C shape, and they get lodged in different parts of your body and can cause stroke and pain and all kinds of stuff. And they were able to deliver gene therapy to patients in two different ways. One, using an episome, or actually it's a lentivirus, which I can go into, and another using CRISPR. So that was actually the first CRISPR example. I think it's Casgevy, is a CRISPR-delivered gene therapy. All the other ones have been episomes, the circular DNA, or this other way I don't have to get into that also inserts DNA into the cell. And you can see the list goes on. And a lot of this is due to the innovation in gene therapy over the past few years. And I'm excited. I think the future is really bright, especially for ophthalmology. There's a lot of diseases where I look at a patient and I say, in 2024, we have nothing for you. But maybe, in the next 20, 30, 40 years, I will have something for them. All right, so that's that one. Okay. Do you have any thoughts about CRISPR or questions? Anything? Like, where do you see potential?
I'm curious. Did you learn this stuff in school?
So I was in school around the time CRISPR was sort of figured out. And so, yes, I learned portions of it. Now, the big implications weren't there yet. We didn't know everything yet. So yes and no. It was still a work in progress. So this has all happened in the past 10, 15 years, a lot of this work.
Yeah. For some reason, this concept is challenging for me to wrap my brain around because I didn't learn it in school.
Sure.
And so thanks for your synopsis.
I hope it helped. I mean, a quick and easy way is we're either snipping with scissors out the bad part and just putting a new thing in, or just snipping it out and letting it heal back together, or we're just giving a whole new piece of DNA for the cell to read. That's the basic idea. So I'm trying to think of other diseases this could impact, but the list is too long to even list on this podcast. So that's that quick medical minute on CRISPR. And let's jump over to another topic. Okay, so I mentioned at the top, brain-computer interface. What is that? Well, it is what it sounds like. Basically, trying to take information your brain generates and meld that with a computer system, so an electronic system, and making them work together. Generally, you're trying to take brain information and deliver something out, like say write an email, or maybe deliver a signal down to a lower part of your body that was damaged from an injury, say a spinal cord injury, so you can walk. But there's also technologies the other way around, where you could take computer information and deliver it to your brain, say an email that comes to your brain, or a signal that helps you not have seizures anymore. So I was going to talk a little bit about that. I think the most popular or most talked about one right now is Neuralink, which I'll come back to. But the original BCI, or brain-computer interface, was the deep brain stimulation, or DBS system, made by Medtronic. This is slide 2. So in slide 2, we're talking about deep brain stimulation. What is this? Well, basically, what you do is you do brain surgery. You go make a hole in the skull and take an electrode, which is a piece of metal, and carefully insert it into the brain down to a specific location near, I think it's near the substantia nigra, which is what's damaged in Parkinson's disease, and deliver a signal in that area to help folks with Parkinson's disease have less tremor and have a better quality of life. As you could tell from the graphic, the electrode is quite long. They have to bolt it to the skull so it doesn't wiggle around in the brain and cause damage. And they kind of separate in space the actual electrode and the electronic system, which is threaded down and placed under the skin on the chest, similar to a pacemaker or what have you, if you've ever had a relative or friend who has these. And this will come back later when I talk more about this. So that's deep brain stimulation. It's been around for a long time, and I can tell you it works in medical school. They would have patients come in with certain conditions, and they had a gentleman who had Parkinson's, and he had a DBS inside of his brain. They had him walk around, talk to us, all this, and then he sat down and they turned it off live in the theater, the classroom, and you could see the effects of turning off the system on him immediately. It was dramatic and quite frightening, honestly, to see how much sicker he could be without this technology. And then they turned it back on, thank goodness, and he went back to how he was. So that was the original one. It's been around forever, since like 90s, late 80s. And now we're in a new era where we want a little more sophistication. We don't just want to send a signal in. We want to collect data out to help people with disabilities due to accidents or diseases so that they can communicate with the world. So at the beginning, I mentioned Neuralink. It's probably the most publicized company. And on slide 3 here, I have pictures of the Neuralink product, which is these little tiny electrodes instead of that big rod that they are carefully inserted into the brain based on all these measurements that they do before surgery so that they can collect electrical information and transmit that into text or what have you.
And you're talking about the one with the penny?
Yes. So actually that...
Which is the Neuralink electrode?
These little hair-like things
okay.
They put in hundreds of these. Actually, no. I forget the exact number. 96. Sorry, they put 96.
So those things
probes.
that look like eyelashes are probes.
Are probes. Instead of a big rod, you have these small eyelash-like things going in, and they're showing here also a picture of a mouse who's had the Neuralink fibers implanted. This is sort of not how it would look on a person, but they're just, this is how you have to work with it with a mouse, and you can see the little unit sitting on top of its head. Next to that, I guess labeled ABC here, is just showing the device that inserts these electrodes. So the electrodes are so fine and so fragile, and the placement has to be so exacting that a human hand cannot do it. A neurosurgeon cannot place this. So they had to create basically a sewing machine like device that puts in the electrode and then carefully backs away so it can go in, deploy the electrode, and then slip out without causing as little damage as possible. And just for comparison, this is how small it is compared to a penny. Here's the big zoomed in view. It's very small, these electrodes. The thing about this is it's very hyped up and talked about, but they've actually never done this in humans. And the recent FDA approval to do human studies is very hazy. What's happening? I looked at the website. It does not say what hospitals it's at. It said it's at IRB or internal review board, investigational review board approval at some hospital. Doesn't say which one. So I don't know what's going on with that. So it's very hyped up and exciting, no doubt, but it's a little opaque about what's happening there with Neuralink. Despite it being the most talked about one, it's the most behind in some ways, although they do have an interesting way of delivering their product. Oh, another thing worth mentioning about Neuralink, and I don't have a picture of this, so I'll just describe it, is the idea is you're not separating the electrodes from the battery and communication hardware like it showed in the deep brain stimulator where it was under the chest. They kind of want everything all there in the skull. So they want a battery and they want the transmission information like right there screwed into the bottom of the skull, and then the electrodes coming down out of that into the brain. Why do they want that? I think it's smaller, more compact, more sleek. The trade-off with that, though, the big concern is that batteries generate heat, and even a small temperature change in the brain can actually melt brain tissue. So there's a concern, a legitimate concern, that this could harm patients because it's too localized. So other folks have thought about this, and another competitor is called Synchron. So let me get their images pulled up here. The image on slide 4 on the left shows where Synchron is placed, which is in the venous sinus, which is the veins around your head, helps drain the blood from your brain. You probably don't think about this much, but yeah, blood goes into your brain, but it has to get out of your brain back to your heart, right? So you have these big veins around your head that help that fluid get away. They're big, they're pretty low pressure, kind of a safer place to do things instead of actually poking brain tissue. What they figured out you could do is go in through veins up into the brain and deploy a coil right there, right in the venous sinus, next to the brain, and the coil is shown here in the picture on slide 4 on the right, that touches the wall. It's like a stent that they use all the time for cardiac stuff. It's a very common design in medicine, and that touches the side walls, which are very close to the brain, and it takes the electrical information from the brain and sends that down a wire that goes down to a chest lead, or like computer system, similar to the deep brain stimulator, a little thing you could see under the skin. So you're separating in space the electrode and the battery and all that. So in theory, if the battery needed to be changed or whatever, you don't have to go do brain surgery again. You just cut into their chest and replace it. Much easier. And the thing about this technology is it works. It's been done in at least 4 folks that had Lou Gehrig's disease, or ALS. These folks have damage to the motor neurons that help you know how to move around. The problem isn't their ability to think about moving. The problem is once they think about moving, that signal doesn't get sent to their muscles, so they can't move their muscles. So this is a perfect because you need a functioning part of the brain to give that signal, hey, move this, do that, type this, in order for any of these technologies to work. You need a working brain in order to send out a specific signal. So in the study, they implanted this for patients, and there's amazing videos on their website on Synchron, synchron.com? Yeah, where you can watch this in real time and see folks write emails, check everything, and otherwise they cannot move. So it's amazing. And last but not least, let me get the final thing. Well, that's pretty cool, but we can skip it. Eyeballs. How has BCI, or brain computer interface, been integrated for eyeballs? So in slide 5 here, I'm showing the Argus 2. Have you heard of this, or have you seen this?
No.
Okay, so the Argus 2, this is for retinitis pigmentosa, the same disease I was talking about earlier with Luxturna, where folks lose the ability to see because, in this case, the outer part of the retina just gets beat up and damaged due to a gene defect. So the inner part of the retina actually works. If you gave a signal to that part of the retina and said, here's some light, here's a letter, whatever, it could take that and shoot it to your brain and you'd be able to see it. So they thought about this and they said, wait a minute, what if I put some sort of sensor into the eye so that the light information can hit the sensor and then it can zap the inner retina cells and tell them that you're seeing something so that they wake up and send a signal to the brain. And so it's a very difficult surgery. And right here, you can see on the far right, there's a picture of an eyeball with this device inside of it. It says electrode array. That's the little photo sensor, basically a little camera that takes the light information and then zaps the cells gently to tell them that they're seeing light. And then off it goes to the brain. Argus 2, as far as I know, I mean, it was done a bit early in my residency time, but now it's kind of fallen off the map. I think perhaps due to the introduction of Luxturna and other therapies that are better and less invasive. So, this is the beginning of something. And you mentioned to me Elon Musk saying he was going to cure blindness. Maybe it's possible. There are ways of thinking about using BCI in other ways to help people see. This is sort of more of a brutish way of doing it, an original way, but maybe with Neuralink, Synchron, something in the future that could be possible. So that's my my medical minute.
Thank you. So when a patient comes in who has the number one cause of legal blindness in senior citizens in the country, which is macular degeneration, and they're basically legally blind, they're going to ask you, Dr. Medert, is there a doctor here in town or a doctor that you know of that is doing anything that can help me? And you know of these technologies, what would you say to that patient?
Well, I would say there's no current FDA-approved study or treatment that involves brain-computer interface for age-related macular degeneration. Again, Argus 2 is only approved for retinitis pigmentosa, and it's kind of fallen out of favor. So at this time, there's no treatment quite like that for those patients. It's interesting, though, because if they don't have atrophy or thinning, they just have little lumpy bumps or drusen that's affecting their vision, you could maybe bypass that with something like an Argus and help them see. But yeah, there's nothing right now.
So you were at Bascom Palmer, which is a world-renowned center for eye treatment and eye research. Were any of these treatments being offered at Bascom Palmer?
Yeah, so Luxturna, the gene therapy where they inject the virus under the retina, was routinely done by Dr. Barakal and Dr. So to answer your question, Dr. Grigori, who's at the VA, but she also goes to Bascom Palmer, had done some Argus, is my recollection that she did that. So yes, they did both. But Argus fell off the map. It just wasn't used as much. It's such a difficult surgery.
Is this Argus or
That's
no?
Argus. So slide 5 shows
Okay.
Argus. It's basically you're doing what's called a scleral buckle, which is like you put a little belt buckle around the eye, and then you have an big bulky electronic case, and then you have this whole piece of hardware that goes into the eye through the sclera and is tacked onto the retina. It's super invasive, very hard surgery. And then they had to wear these special glasses that are also shown here in the image that helps transmit the light information. And just make sure everything works together. And then there's a processing unit. It's really complicated, and the patients didn't see as well as the risk is worth. It's a tough cookie. It kind of fell out of favor.
Okay.
But it's iterative. I think all of these things are the beginning of something.
Yes.
I don't think this is the end. I don't think Argus 2. I guess there was an Argus 1. I don't know what that looked like, but there's hope here. I just don't know when that hope will manifest.
Got it.
Yeah. All right. So that's our medical minute. A fan favorite, I can already tell. So that's this episode. Next one will be more of our long form. We should say, or slightly more long form version. We should say our famous catch line. Are you ready?
Ready.
Okay. Thank you for seeing eye to eye.